Swami the Teacher YouTube channel. Today we are going to discuss a lesson of the grade 10, that is the 14th lesson, the continuity of life. In this continuity of life, we have to discuss about two main parts, that is one is the continuity of plants, the other one is the continuity of animals. In that one, today I thought to select the continuity of plants which is done by the vegetative propagation. That means the plants when they are doing reproduction. So reproduction is a process to make their generation continually in this world. So producing of the offsprings by the existing generation is known as reproduction. Plants also do reproduction through mainly two ways. One is the natural reproduction, the other one is the artificial reproduction. Today we are going to discuss what is this artificial reproduction. We called it as vegetative propagation. Propagation means giving another generation. With the interference of the human, this one is happening. Mainly the vegetative propagation can be divided into four parts as stem cuttings, layering, grafting and tissue culture. Stem cuttings is common, especially with our mothers. Whenever they go for a neighboring house, they just bring some stems and the branches of some plants. And through these branches or through the stems of this plant, when we just keep them under the soil or when we just plant them under the soil, a new plant can be taken. Especially the roses, shoe flower, croton, those plants, they do the propagation through stem cuttings. The other method is the layering. Layering, we need some sort of a technical knowledge to do layering. Layering can be mainly done in two ways. One is known as the ground layering. The other one is called as aerial layering. When we talk about the ground layering, in this one, we specially select the plants who has the branches very close to the soil. Then when a plant is having a branch close to the soil, if this is its soil and if this branch is somewhat near for the soil, what we do, we just make a small cut in the underground part of this branch. You can see there's a small cut and we cover this one with the help of some soil. Here is the soil, so we cover this place with some soil and some compost medium. Then after some times, you can see that the rootings have come from this place. New roots have occurred. After that, you can just cut this branch from this place and you can plant this one in a separate area. So the main advantage of this one is in the layering, we do not separate the plant for layering from the mother plant. While it is attached to the mother plant, you can do the layering. So this one we call as the ground layering. Especially we use this one for the lemon plants, especially for the lemon plants. Then there is another one which is called the aerial layering. So when we talk about a plant, all the branches are not close to the soil. Some are in the top of the plant. So for the branches who are in the top of the plant, we can do the aerial layering. So if this branch is near in this way, so what you can just do is, you can just remove the ring of this bark. We just remove the ring of this bark and we keep a compost and soil layer to this place. We just cover that, the ring where I removed the bark by using compost and soil mixture. Then after sometimes, after about few days, you can see the roots are coming because this is a very good nutritive media. So the roots are coming from this bark. Then also you can separate this plant from the mother plant and just grow this one in a separate place. So this procedure is the thing we call as the layering. Layering can be done in two ways as the ground layering, the stems which are very closer to the soil, and aerial layering for the branches which are somewhat higher from the soil. The third method, that is known as grafting. So when we talk about grafting, grafting also has two methods as bud grafting and twig grafting. Bud grafting and twig grafting. When we talk about this bud grafting, these grafting plants are now very much common in our society. In the bud grafting, especially we use this one for our rubber plants. First you have to select a bud, a apical bud, lateral bud, especially near these leaf scars you can find a bud. After taking this bud, you need some sort of a very good knowledge for this one to just select this bud. To select the relevant part with the bud, you have to use a budding knife for this, and also you must have some sort of knowledge of taking that one out. Without damaging this bud, you have to take this out. Then what we do, in the case of grafting, we are going to just mutate or join the two characters of two plants. One plant is giving the shoot system and another plant is giving the root system. So two parts, two plant parts, we are just combining these two to get a very good variety of fruits or very good characteristics. So the part which gives the root system, the plant part which gives the root system, we call this one as the stalk. This is the stalk part. So main features we are looking from the stalk is it should have a very good root system, very spread root system, and it should be withstanding to the environmental conditions, especially the drought conditions, as well as it should be free from the root diseases and the pests who are just damaging the roots. The other part which we are connecting to this part, which has the shoot system, is known as the scion. So shoot system is the one who is giving the fruits or the foods for us. So definitely this scion should give a very good harvest. It must give large number of nuts, or very huge number of nuts should be given by it. As well as it should have a very good leaf system. The branches and the leaves should be very good because they must do more and more photosynthesis. As well, it also must be free from the diseases and disorders. By considering these factors, we select a stalk and a scion. Now we are going to combine these two. But for the combination, always you have to select the plants from the very same variety or very close varieties. That means you can't just combine a rambutan plant to a mango plant. But in the mango, we know that there, so we can combine Vilat or Kartha Columban because they belong to the same species. We can combine their characters together. So now we are going to combine this one. That can be done in mainly two ways, as the bud grafting and the twig grafting. So when we talk about the bud grafting, first of all, the scion is coming as a bud. This is the scion. And the stalk we select from the root system. In here you have to make a cut. You have to make a cut in this place to keep this bud. So these cuts can be made in different shape. T shape, then in the H shape, or else you can use a V shape. These cuts can be done in different shapes. So you have to just make this shape, and we are now going to place our scion insert that one into this cut. Okay. Then I am going to keep the scion here. These two cambiums are now getting touched. Then what you have to do, you have to wrap this place from the ground to upwards. From downwards to upwards, you have to wrap this place by using some sort of polythene. Otherwise, if it rains, the rainwater will go here and they will just spoil our grafting. Then after about 2 or 3 days, you have to again remove this part. Just open. Just let them but to open outside. And again cover from this place. Just open only this area. Then sometimes after about a week, you can see a new branch has emerged from this place. A new branch has emerged from this place. Then you can just cut this plant at about 15 centimeter area. You can just cut this one, and then this part is removed. The top part of this stalk can be removed, and you can get a new plant from this way. So this is the procedure we are doing in a bud grafting. So it should be very much attention. You have to carefully pay your attention to select the scion, how to get that scion, and making of the cut in the stalk. That is about bud grafting. Then there is another one, that is the twig grafting. So when we talk about the twig grafting, in that one we use two stems. Two stems are used. This one is coming from the scion, and the other part comes from the stalk. So this is the stalk, and this is your scion. So we just make their cambiums to get contact with each other. So this twig grafting is somewhat difficult to done in the monocotyledon plants because they do not have a cambium. Especially in the dicotyledon plants, we are doing this method. So when we are doing this grafting, again you have to wrap this grafted place with a polythene. So after some times you can see this place has get connected very well. If the place has get connected very well, you can just remove this polythene out. This is very much common. Now there are twig grafted plants are grown in our home gardens also. But the main disadvantage of these grafting plants is their lifespan is very short, as well as they are giving the harvest only for a very small period of years. As well as we can't get any woody value, just like our older jack trees, mango trees, we are using them to make the furnitures. But these plants you can't use to make furniture because they don't have a very good wood value. Then the last method is the tissue culture. When we are talking about tissue culture, that is the advanced method when we are comparing from these four. Tissue culture is done in a laboratory. That means the growing of the plant parts in glass bottles. In the sterilized and controlled environment, we call it as a tissue culture. So first of all, you have to select a part from a plant. We think we are just taking this apical bud. Normally we use the buds, apical buds, lateral buds or root tips are used as the callus here. Then we use this part and we put that one into a petri dish. This petri dish is full of a nutritive media. How to make this nutritive media? To this one we put hormones, growth hormones, sucrose, glucose and some minerals, vitamins which are needed for the growth of a plant. We provide all those things and we just mix that one with some agar and we can make this growth medium. Then we put this tip inside to this growth medium. We are putting this tip to the growth medium. Then after we are putting small parts of this one, small part of this bud to the growth medium. Then after about some times you can see the rootings and the shoot system. The root and the shoot system is now appearing from this one. Then we separate these plants into test tubes. The plant is put to a test tube. These plants are separately put into different test tubes. Then we let to grow them inside the laboratory. It should be a sterilized condition. There should be no microorganisms. Otherwise the microorganisms will grow on this agar medium. We must prevent that one. As well as you have to provide all the nutrients needed for the growth of this plant artificially. And the lights are provided to give just as the sunlight, we have to provide the light. And most of this time, these tissue culture laboratories are air conditioned. Then this plant will grow into plantlets. Then this one will grow. After sometimes it will grow into a very good plantlet, having the leaves and having a root system. Then since this one has grown in an artificial environment, we have to just practice this one, or we have to just that means we have to just allow that one to grow in some controlled environment. Then we can just allow them to grow in a controlled environment, just like a net house or inside a polytunnel. Then after with this become 2 to 3 weeks, you can just expose it into a normal environment. So these are the major vegetative propagations. That means the way how we can reproduce another plant with the help of the human artificially. This is the methods. These are the main 4 methods we are using. So this method is very advantageous for the plants that can't produce seeds, because flower and the seed is the main reproductive part of the plant. But if they can't produce that one, so there will be no reproduction, then their generation will be over from that place. To avoid that, these methods are very much useful for the plants that which can't produce the seeds. As well as through this one, you can see we can just make a new plant within a very short period of time. Especially when we talk about tissue culture, we need only a very small petri dish. Within a very limited area, you can make number of plants. But when we talk about the disadvantages of this vegetative propagation, they have a very low lifespan and technical knowledge is needed. This one is somewhat costly. So this is the way how we are doing the vegetative propagation for the plants. We will again meet with another video which contains the natural propagation of the plants. So thank you for being with my YouTube channel. Thank you very much.